Okay, good evening, everyone, and welcome to the first seminar of the 2024 edition of Orizzonti Medicina and Biologia. I'm Antonio Raffaele Citarella, and here's my colleague Flavio Delisio, both second-year students of Medical Sciences at Scuola Superiore Sant'Anna. Before we begin, I would like to provide a brief introductory overview of our institution. Scuola Superiore Sant'Anna is a public university that admits students through a merit-based selective process. Students at Scuola Superiore Sant'Anna have the opportunity to personalize their academic path in a multidisciplinary environment that promotes personal and academic growth. Despite the young age of the institution, founded only 36 years ago, it has achieved a high reputation internationally, both in terms of education and research, ranking among the top Italian university and the top 15% globally in the recent Times Higher Education World University ranking. Our institution is constantly evolving and offers students access to the latest discoveries and research through collaborations with prestigious laboratories and universities around the world. The school is committed to providing an integrated medical education spanning engineering, biotechnology, economics, ethics, and other disciplines, offering students ever-changing opportunities in an interdisciplinary context.
The vibrant cultural environment inspired the creation of the seminar series entitled Orizzonti Medicina e Biologia, which allows us to welcome brilliant researchers engaged in medical discovery and to share their experiences and life paths. We would like to take this opportunity to mention that this is the 12th edition of this seminar series, which has continued to grow thanks to the commitment and passion of all those who have contributed in the past and continue to do so in the present, involving an increasingly large and diverse audience. Before we begin, we would like to share with you a small summary of the last edition of Orizzonti through this video. Thanking Professor Michael Salter for accepting our invitation, I leave the floor to Professor Vincenzo Leonetti, the one in charge of this project, who will introduce the guest speaker. I thank everyone for your attention and wish you a good continuation of the evening.
Good afternoon and welcome at Scuola Superiore Sant'Anna. It's a pleasure to have here Professor Michael Salter. Michael Salter is a great scientist and his research is mainly focused on a difficult issue, is the pain. And just some news about him. Michael Salter got his degree in medicine in 1982 at the University of Western Ontario in Canada. And in 1987, he received his PhD in physiology at the McGill University in Montreal. From 1987 to 1990, he was a postdoctoral fellow in Canada at the Playfair Neuroscience Unit of the Toronto Hospital, and then at Mount Sinai Hospital Research Institute in Canada. And from 1991 to 1995, he was assistant professor of physiology at the University of Toronto, where he's still conducting research activity as a professor of physiology and member of the program in neuroscience. After this period of spending in the lab to grow his skill and try to improve his knowledge, started as an independent scientist in 1990, and his department was the Department of Neuroscience in the Hospital for Sick Children, where then he was the head of the program of neuroscience and mental health till 2015, and the director of research, and actually is the emeritus director of research in the same institution, and actually is still conducting research in the pain physiology and pathophysiology. Professor Solter is a renewed scientist because he tried to change the paradigm, investigating the crosstalk between different cells in the nervous system, and even trying to understand the molecular mechanism underlying the tolerance and sensitivity to pain, even to neuroplasticity. He published his finding on prestigious journals, some example, Nature, Science, Cell, Nature Medicine, and Neuron. His studies are at the fundamental of understanding neuropathic pain, that is a really difficult issue, and is also a difficult disease to treat, actually. He gave a great contribution to other diseases like stroke, neurodegenerative diseases, schizophrenia. And thanks to his achievement, he was recognized with different and several prestigious awards. Some examples are the International Research Scholar at the Howard Hughes Medical Institute, and even the Fellowship of the Royal Society of Canada, and even the Romack Seller Family Neurobiology Innovation Award of the Sickkids Foundation. He had also some politically relevant role he played in the governance and nominating committee of the Governing Council at the Canadian Institute of Research. And since 2010, he's a scientific advisor of the Canadian Pain Coalition. He also had a great collaboration with the industry, the drug company. He founded several drug companies, and he tried to develop different drugs and relevant drugs. He contributed to development of the neranetide, and is a drug that tried to reduce the injury following a stroke. And actually, he published more than 200 original articles, and he has an H index above 78 from Scopus. He signed the 14 patents, and he is actually acting as reviewer of prestigious scientific journal like Nature, Cell, Science, and it is also in the committee of grant institution like NIH, NCS, and even Telethon Foundation. So thanks to Professor Solter to be here, and I invite you to come to the podium and start your lecture. Thanks.
Grazie, D'Angelo. Buonasera. So I'm going to start with a little reminiscing because it's been a wonderful visit here. Thank you. Gotcha. Okay. So I'm here today primarily for a couple days to visit my very dear friend, colleague, and former boss, Flavio Cocciani. Flavio was head of department and I started at the Hospital for Sick Children, and I very fondly remember him for his wise counsel, his friendship, and just for being a superb human being. So Flavio, thank you for everything. You made my life at Sick Kids so interesting and important, and I learned so many things from you, most of which was be polite and be a good person. So thank you, Flavio. Okay, so let's give this a go. If I can, let's see. Are you gonna go? No.
That's fun.
Nope. Doesn't. Let's try this again. Okay, that's working.
That's fun. Use
That's the laser.
the last one. The last one is the, okay.
Do you want me to take the mouse? I can just use the mouse.
Yeah.
Or the keyboard, that's perfect, yeah.
Person space.
Let's try this. Spacebar. Nope.
È il bello della diretta. Okay, now it's working.
Okay. So this is important in medical schools today that although I have been involved with several pharmaceutical companies and we are still trying to develop a stroke drug, nothing that I'm going to tell you relates to any of the work that I've done for or with industry. Okay, so what I'm going to talk to you for, what did you say, Vincenzo? 2.5 hours. That's right. Okay. Okay. Grazie. Okay. So I was telling my student hosts this afternoon about my interest in pain, which really developed as a second-year medical student at the University of Western Ontario. And I was fascinated by the whole concept and the mind-body issues and the philosophical issues raised by pain. And so this has been my life's work and passion ever since then. We're still working on it, so we haven't solved things yet, but we're still working very much on it. And so it's the kind of pain that we're interested in is the kind of pain depicted in these kinds of art pieces that I took unceremoniously from the web over many years. This is the kind of pain that disturbs people's life, that really affects their quality of living and affects their relationships with their friends and families, their ability to work and to think. And Vincenzo and I were talking about this before. This is what we're trying to, if not cure, at least put a big dent in. And so the approach that I've used to do that, which you guys can think about as students, is to devote my life to doing research with the hope of bringing forward new therapies and potentially cures for this. So these are the kinds of things that continue to obsess me about this pain that separates individuals and causes great suffering and anxiety. This is what we're trying to model in experimental animals, and then we're trying to learn something about the molecular and cellular pathways that are involved in this so that we can change people's lives. That's what we're trying to do. So just to give you a sense about the problem, these are numbers. This is, even though it's 2011, these numbers I don't think have changed too much, and there have never been more accurate numbers, but the numbers of people around the world is always roughly thought to be with chronic pain. About 15 to 20% of people, especially us old white-haired people, right? As you age, the prevalence of chronic pain increases quite dramatically. So there's lots of numbers on here about costs and dollars, and we could convert those to euros, I suppose. But for me, the biggest thing is that is what's not on this slide. And Vincenzo, don't strangle me, but the disability caused by chronic pain is greater than that caused by heart disease, and cancer combined. So a huge, huge unmet social and medical need that there are many, increasing number of people around the world are working on. So I'm going to give you my perspective on this. So first, I'll give you a little bit of background to bring you up to date, and primarily directed towards especially the young students about the way that I think about pain, and then two vignettes about that, about a new model that we've developed, and then about an attempt at drug discovery from one of my students. So this is the way the field, not just me, thinks about acute pain. So it's that pain that comes about directly associated with injury. And if you think about this from an evolutionary perspective, all organisms, whether they're big or small, need to have ways to detect and respond to tissue damage or potential tissue damage. And that provides both the ability to repair any damage, but also provides a learning signal that all of us use to know when to stay away from certain things, like a hot stove or the pointy end of a needle and things like this. So this is absolutely essential for survival. The experience of nociception, and in human beings, at least, we experience this as pain. The flip side of the coin is what we're concerned about, is this non-adaptive pain that comes about, may often be the result of an acute injury producing chronic pain, but then even after the tissue healing occurs, that the pain continues and produces this suffering and distress that I was showing you in the images. So this is what the major problem. There are, in most cases, very good pharmacological and non-pharmacological means to address acute, particularly acute postoperative pain. But the big and unmet need is around chronic pain, and that's what we've been focusing on. So what we've learned over the last few decades has been that we've been working on this idea of developing a new biological understanding about pain and pain mechanism. So what we know now is that chronic pain isn't just the same as acute pain, except it lasts a long time. That there are many different biological mechanisms, and that chronic pain is undoubtedly a series of diseases, if you will, or some people would say mechanistically distinct disease processes in response to various situations that arise, particularly injury, but not always injury. Sometimes chronic pain can come about without any obvious or initiating injury. So this is what we're trying to do, is to try to think about the mechanisms underlying chronic pain and how we might be able then to address those, mostly pharmacologically, from the way I think as a person trained in math and chemistry. I think about molecules, and molecules in this context means pharmacological intervention. So just a couple terms, because I'm probably going to come back to these. So neuropathic pain, which is the major of chronic pain categories. This is the one where there's the most unmet need, is around pain that comes about in response to injury or damage to the central or peripheral somatosensory nervous system. And in many places along the neuraxis, there are areas where damage causes pain hypersensitivity, and is characterized by these three cardinal signs: allodynia, which is a painful experience in response to innocuous mechanical stimuli; hyperalgesia, pain being more intense than you would expect on the basis of the size of the noxious stimulus; and then spontaneous pain. And we have reasonably good models. We can argue or discuss about whether these have utilitarian value or not. But for allodynia and hyperalgesia, spontaneous pain is much more difficult to even conceptually model in experimental animals. So what the data that I'm going to show you relate primarily to our and the field attempts to model allodynia and hyperalgesia. So for those of you that don't think about the pain system all the time, which is undoubtedly all of you, other than me, that we know a lot about the peripheral processes that are involved in pain hypersensitivity. All of the primary detectors have been identified. They're largely proteins that detect things like protons or inflammatory mediators. These are transformed into action potentials which travel up primary sensory afferents and interface with a nociceptive network in the spinal cord dorsal horn that we've attempted to talk about here. And then the pathways, neural pathways on this have essentially been completely identified long time ago in terms of the ascending pathways that send information up to the brain. In the field, we have a saying, which is, it's not pain until it's in the brain. So it's that complex experience that we have that is mediated by a variety of different neural networks that span large areas of cortex and subcortical regions. And we've known this, and we've been working out the details about this for many decades now, the fundamental nervous, neuron-based aspect of this. Now, in showing you these, I picked these 5 examples. If you look at the dates, they're all from last millennium or the beginning of this millennium. Now, most of, at least the first year students, were not even born. Some of them were. And I picked these on purpose because at that time, in all of these, what you see is neurons, 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 and more neurons. And I'm going to give you a slightly different perspective that has certainly changed my mind over the last 24 years about it's not just neurons, but it's the immune system and neuroimmune signaling. And if you think about it for a while, or if I was to think about it for a while and think about what are the bodily systems that are involved in detecting and responding to tissue, damage or potential damage, for me as a neuroscientist, that's neurons, the nociceptive neuron system. For immunologists, it's the immune system. Who cares about the nervous system? This is the job of the immune system. And obviously what we've learned over the last couple decades is it's both systems that are involved in this. And it's the critical interplay between neurons and non-neuronal elements in the periphery and in the central nervous system, and immune systems that are even systemic in some cases. So this is what I'm going to try to give you a hint about, and give you mostly for the students, because we were talking before about career trajectory and how do you get to various places. And so to give you a little bit of the feel for what it was like as we started to move through these things just over 2 decades ago. Okay. So the first part of the talk is really about microglia, which when we started were considered housekeepers in the central nervous system. They were non-excitable cells. So for the vast majority of neurobiologists, including me, non-electrically excitable, not interesting. Okay? These are immune cells, as my good friend Clifford Wolf told me once. These are pus cells. Why would we care about pus cells in the nervous system? That's what he told me. But as we and the field has learned over the last couple decades, these are very interesting cells that are involved not just in response to localized damage, but have many physiological roles in development, in synaptic plasticity. And these are cells that are surveying the brain continuously, the brain and spinal cord continuously. So these processes here that you can see when they're in what used to be called the resting state, when we only had static images, what became apparent with video movies of microglia over time is that these cells are continuously extending and retracting their processes constantly. And the estimate is that the entire extracellular volume of the central nervous system is surveyed by microglial processes every 12 to 24 hours. So these are cells that are involved in vigilance and also other processes that are critical for development. Okay, so none of this was known when we started on this journey as we went along. So just to give you, because you're going to see a number of data slides, and I know you're not used to this. So we're interested in peripheral neuropathic pain, so pain that's driven by injury or trauma to a peripheral nerve. We can model this in animals in a variety of ways, for example, by taking branches of the sciatic nerve and cutting, crushing, burning, twisting these, and that gives characteristic behavioral change where the animals on the side of the injury become much more sensitive to otherwise innocuous mechanical stimuli, right? So a model of allodynia, and they become much more sensitive also to over-threshold stimulation. But typically we use sub-threshold stimulation where the animals show hypersensitivity. So, we see that here in terms of the threshold for the animals to withdraw to mechanical stimuli applied to their hindpaws. So this is the thumb of Makoto Suda when he was a postdoc in my lab. Makoto will play majorly in the next few slides. And this is where he would put, there is a thin filament here that you probably can't see. I can't see it from here. But these animals, after, in our case, cutting and ligating two of the three main branches of the sciatic nerve, these animals become so sensitive to mechanical stimuli that when I apply that thin filament, the so-called von Frey filament, to the glabrous skin on my hand, I can't even feel it. So maybe that's just telling you how old I am, but maybe that's telling you a little bit about how sensitive these animals become. And they show what we call nociceptive responses. So it's not, we're often criticized, the field is often criticized for using these models because they are reflexes, but the animals attend to the paw as if it was hurting after this kind of intervention. So, and there's lots of reasons to think that it's certainly a highly regulated reflex. Okay, so if you didn't follow that, just more pain is down here, less pain goes this way, okay? So that's how you can interpret the following graphs. If anyone asks, I never told you that in that way, okay? So, okay. So one thing that we found early, well, was in the field for, I would say now at least 40 years, is this observation that after peripheral nerve injury, like I told you about, there is a characteristic response of these cells, which generate this pink color here. It's a little bit hard to see, I think, but the pink color comes from cell surface labeling of microglial cells, which normally look like this, sort of like the one I showed you in the other slide, where they are highly ramified and in this surveillance mode. On the side ipsilateral to the injury here, you can see that the shape of the cells has changed, their number has increased, and they look like they're kind of spoiling for a fight, right? So this is, they go from a state of surveillance to another state, which we're going to start to call the P2X4 receptor plus state, okay? And when I say injury side here, it's not that the injury was here. The injury was somewhere halfway to the tower, right? At this scale. So action at a distance that the microglia are detecting and responding to this injury of a peripheral nerve a long way away. So as I said, this was an observation that when we go back, we see in the literature summarily ignored by 99.99% of the neuroscience community, including me, right? Pus cells, who cares about them? Okay, we were kind of, as you'll see in the next few slides, we are dragged kicking and screaming into this field. So we were interested, we, meaning me and my collaborator, Kazu Inoue, who really started this project in his lab in Tokyo. We were interested in purinergic receptors, and particularly as the field had evolved from the time when I was a graduate student working on purinergic receptors in the cat spinal cord, the field had evolved to multiple different receptors that you could activate or inhibit with various ligands. And so we started by trying to get a sense of, it's a very simple question. Are purinergic receptors involved in maintaining this hypersensitivity after a peripheral nerve injury? Very simple. We had crude pharmacological tools, but they were certainly better than the tools that had been available even 5 years before that. So the way that you read this slide then is, here's the change in withdrawal threshold. So pain this way, and then we can reverse this hypersensitivity by injecting intrathecally. So this means at the level directly around the spinal cord, in this case at the lumbar level. So we inject this compound TNP-ATP, which is a blocker of a specific subset of purinergic receptors, and we reverse completely the hypersensitivity. Single injection, the reversal reverses, so we go back. So it's not that we injected something that killed all the spinal cord so the animals couldn't respond. They can respond just fine. We had some crummy pharmacology at the time and a little bit of old school siRNA that we could use to identify P2X4 receptors as the receptor that was mediating this hypersensitivity that we could reverse with TNP-ATP. Okay, so that, if you were a purinergic person at the time, that could be moderately interesting. It was fabulously interesting to Kazu and me, but, you know, mostly not so much. So the interesting part came when we started to think about what cells are expressing these P2X receptors that are involved in maintaining the hypersensitivity. And we sort of went through the litany down the list. Neurons? No. They don't express P2X4 receptors. Astrocytes? No, they don't express P2X4 receptors. But I will tell you, you know, we were talking about career earlier today. I will tell you, when Makoto Suda showed me this image here, my life, and the life of my lab changed in an instant. Because, like it or not, it was those pus cells that were expressing P2X4 receptors, the microglia. So this all colocalizing with the microglia marker. So the logic flow, and this is where I think that one of the most important things in science is to follow the data to the logical conclusion that you get, no matter how much you don't like it, right? Because I hated this conclusion at the beginning, right? So the logic went, P2X4 receptors are necessary for the hypersensitivity following peripheral nerve injury. P2X4 receptors are expressed exclusively by microglia. Therefore, as much as I hated it, microglia are necessary for maintaining this response. So that started a whole series of things that we've been following for the last 22 or 23 years, actually. Okay, so that's where we were with that. But immediately, once we had seen microglia, we knew that the microglia weren't the whole story, because neurons are actually important in this response. There's neural networks that have to be activated for the animal to respond and to move its paw. So there has to be, as we knew from the beginning, some sort of communication from the microglia to the neurons. And the answer to that came through this paper, which was by my office mate as a grad student, Yves de Koninck's lab, published in the very next issue of Nature that year, way back all that time ago. And Yves had been working on inhibitory neuronal processes and identified this very interesting way of limiting inhibition in the spinal cord. And it was through knowing that most fast inhibitory transmission in the spinal cord is mediated by GABA or glycine through activation of a chloride ionophore, which meant that to have this inhibition to be as powerful as possible, you needed chloride to be low, and the way chloride was kept low is through KCC2. So what Yves and Jeff Kuhl had discovered was that in situations like we used in the microglia part of the story, that they cause peripheral nerve injury, that what they found was that there was a loss of KCC2, chloride went up, and that was producing loss of inhibition in this way. For us at the time, who didn't know about this story until it got published, we thought of this as a rather strange way to produce inhibition. You might think about blocking GABA receptors or changing the number of GABAergic synapses. This was an interesting and provocative way of thinking about it. So then a couple of months after this, Eve and I got together at a neuroscience meeting, and I think each of us had the same thought. Maybe your story is my story, and my story is your story. So we got together, and through the tireless work of a number of people in each of our labs, we were able to come up with the following, which is that in the naive state, KCC2's on, chloride's low, GABA's powerfully inhibitory, and the microglia are in the surveillance mode, right? So that's one part. And then after, the microglia are intermediaries in causing the decrease in KCC2 by being activated and expressing P2X4 receptors, releasing brain-derived neurotrophic factor, which is the mechanism through which KCC2 goes down, chloride goes up, and GABA becomes less inhibitory. So we had this as our fundamental molecular mechanism since 2004 about. Okay, so that's been the idea that we've been following for quite a while. And over the years then we've refined this quite a bit. Turns out it's not just in a loss of inhibition by KCC2, there's facilitation of excitation that's mediated through NMDA receptors. And then we've been working our way through the molecular pathways by which PDNF is synthesized and released, and some of the interactions here and how TrkB signaling decreases KCC2 and enhances NMDA receptor function. And as we've gone along, then we realized that this part of the mechanism is also involved in inflammatory pain hypersensitivity, which is in our hands completely independent of microglia, mediated by tissue inflammation, not by cutting peripheral nerves. That a very distressing situation called morphine-induced hyperalgesia is dependent upon co-opting this mechanism from here to activate the release of BDNF and kick off this signaling cascade this way. And we filled in a little bit on this side about what is the linkage between TrkB receptors and NMDA receptors, which is largely what the other half of my lab works on. So we had this model in mind, and we kept working on this for a long, long time. So as we went along, then I'm going to make a very long story very short, which it turns out that all the experiments that I showed you, at least from the nerve injury side, done in my lab, in Kazer's lab, in Yves' lab, and many of our collaborators' labs, were all done in male mice, to a person, to a mouse, or a rat. So we were prodded by a friend of ours, of even mine, called Jeff Mogul, whose part of his work was on finding sex differences in various processes and pain in particular. And so we did a whole series of experiments with Jeff to attempt to disprove that there was a sex difference, and we failed in that. There is a sex difference in this. Oops, let's see if I can get this to go again. There is a sex difference in that this pathway, which we had meticulously dissected in males, is not on in females. So that females actually inhibit this pathway, and that they signal from a set of cells, which I told the students this afternoon are probably T cells, and Tuan Trang, who's been involved in this for a long time, has discovered what this subset of cells is and what this mediator is, but he won't tell me. He's trying to get a paper into one of the cell journals, and he's not going to tell anyone until it's accepted. But basically, this part of the signaling mechanism is intact in females, but this part, the microglia part that's on in males, is off in females, and there's a different neuroimmune signaling mechanism involved in females. So that's where we're at, essentially, on this part of things. We've been, again, working out more and more meticulous details about how the signaling in the neurons works. But for me, the function of this is really around an immune or glial part interacting with a neural part. Okay, so for the rest of the talk, just remember immune and neural as the two key nodes in this. And then there's a lot of details that we've worked out and other people have worked out in all of those. Okay, so I think the field, us and many other people who have picked up on microglia, have really been very successful in promoting the idea that microglia are important. And one index of that is the number of citation or the number of papers that have microglia and pain somewhere in the article. And this is just the number of papers per year published. This was us, here, 2000, and things have just continued to accelerate. So I think it is true that it is hard to go to a pain meeting, at least a neuropathic pain meeting, without the mention of the word microglia. Okay, so from that part, it's been massively successful, except there have been several trials of microglia drugs in, I think, the wrong pain patient population selected, but that's just my opinion. These drugs haven't worked. So maybe there's more to it than just neuron microglial interactions. And that's what I'm going to tell you in the next little bit. Okay. So as I said, there was 3 parts to the talk. The first part was the introduction to get you where we are. The next part's going to be on a microglia independent form of neuropathic pain that we've identified. And then I'm going to spend the last few minutes telling you about what I think is a cool project on using RNA sequencing for potential drug discovery. So we got into this because I had a neurosurgeon who saw patients with neck disc herniation primarily, and we were trying to come up with a model. And I suggested to him that maybe we should take the disc material, because he had this idea that disc material might be what's causing a lot of pain in individuals with herniated discs, particularly in the neck. So I suggested to him, well, let's take advantage of the power of mouse genetics and see if we can learn something of this, which means we have to have a model that's reliable. Well, first we have to know whether this disc material actually causes hypersensitivity. And then we need to have a model that's relatively innocuous to the animals. So the suggestion was, let's see what happens when we take the nucleus pulposus material and put it in the wrong place, which is gently along the sciatic nerve. And when we did that, we produced a mechanical hypersensitivity that lasted about a couple weeks and then spontaneously reversed. And along with this, then we produced cold sensitivity, we produced heat sensitivity that was enhanced, and we produced a small motor deficit in these animals because we were putting this on the sciatic nerve, which of course is a mixed nerve. So all of these phenocopy precisely what happens with traumatic peripheral nerve injury, right? So mechanical hypersensitivity, cold hypersensitivity, heat, and a little bit of motor effect. So we were thinking then that this is, now we know we're putting the nucleus pulposus material right beside the nerve. So we're by definition interfering with the somatosensory nervous system. So to me, this is neuropathic pain, at least neuropathic mechanical pain hypersensitivity in the rodents. Okay, there's lots of kinds of experiments. And one of the rules, just for the students, one of the things that I try to convince people in my lab is that control experiments are much more important than the actual experimental experiments, right? So the rule should be, in my view, 3 control groups, 4 control groups to 1 experimental group. Now, we don't do all that all the time, but we try. So one of the obvious things is it just sticking a mass there gently placed beside the sciatic nerve that's causing this? So we came up with a number of different ways to address that with sort of inert materials that we could make the same size, and we could put them beside. None of those produced hypersensitivity, just the nucleus pulposus material. And was it really that we needed to be close to the nerve with this nucleus pulposus material, or would sort of anywhere in the thigh, which is where we put the material in this part of the sciatic in the thigh, would simply somewhere in the thigh be good enough? So we use the nucleus pulposus into the muscle a few millimeters away, didn't do anything, whereas the nucleus pulposus still produced the sensitivity. So it seemed like we could produce something that was at least phenocopied behaviorally, the hypersensitivity, and the obvious controls didn't do anything. So we were buoyed to continue on with this approach. Oops, you don't want to turn again. Let's see, I'm going to go backwards. Nope. Okay. Doesn't want to go.
Okay.
Perfect. Okay, so before we got too far into this, we wanted to know, is this a male or female thing? All the experiments, the initial ones we did in males, because at that time we were definitely not sensitized to this issue of male-female differences in mechanism. So essentially everything that we saw in males, we saw in females. So at this stage, we have no evidence for phenotypic differences or mechanistic differences in this model between males and females. So we've continued on with this. And when we got this set of observations, I thought, oh, okay, we've got something that we're interfering with the sciatic nerve. It's going to be the microglia story, and no problem. We just go, we'll finish this, and then we'll be able to use this as a slightly different way of producing neuropathic pain hypersensitivity. Now, the problem, oops, doesn't like me again.
Try again.
There we go. So you saw those lovely pictures of the pink. This is all green. So this is our sham control animals. You see the microglial cells lovely distributed around the cord. The neuropathic pain or nucleus pulposus, no obvious, this is the ipsilateral side here, no obvious increase, a tiny little increase, which we know comes from the skin incision. And compared to this in spared nerve injury, which is the name of the model that we use of traumatic injury. Okay, so that got us thinking, oh, there's not really a microglia response, but we knew that, at least in terms of the shape of the microglia and the number. But we knew shape and number really were not predictive. We could interfere with a lot of different pathways, parts of the pathway. The number would keep the same, the shape would keep the same. We could eliminate P2X4 receptor downstream, BDNF blockers, all these things. So we knew that you didn't have to have this increase in number of microglia, nor the shape change. They were not necessary for the pain hypersensitivity. So, but we thought this is a bit unusual. So we went back to a number of different approaches that we'd used in the part of this, the 15 years of the story that had led up to this point. One was, as much as I hate to use this drug because no one really understands how it works, but it works well to prevent, to reverse SNI-induced hypersensitivity. Absolutely no effect when we gave this compound in the animals with the nucleus pulposus-induced hypersensitivity. And we had developed BDNF knockout mice as part of the proof that BDNF was coming from microglia in the first part of the story that I told you. So we used those mice, and there was absolutely no difference in the behavior of the animals, whether they had BDNF knocked out in the microglia or not. So mechanistically, then, this had become quite different for us and got me really intrigued because, I'd gone from what are microglia to microglia are the center of the universe when it comes to neuropathic pain to, maybe not so much. So we kept going on this. We're thinking, are we doing something wrong? So we thought this is just a picture of the nucleus pulposus and the part of the sciatic nerve. So there isn't obvious compression or swelling or anything around the nerve. And one of the characteristics of traumatic nerve injury is that in the cell bodies of the neurons that are damaged, there's large transcriptomic changes. And one of the major proteins that changes is this protein ATF3. And so you can see that in spinal nerve injury or this chronic constrictive injury compared to the naives or shams, which have very, very little ATF3 expression. There is dramatic ATF3 expression in these traumatic models, but in the nucleus pulposus by the sciatic nerve, really nothing that's statistically significant from the sham. So again, we're not producing obvious compression or traumatic injury to the nerve, and yet the animals have pain hypersensitivity, the characteristics of which are indistinguishable from the pain hypersensitivity produced in either of these models. So now I thought we're really on to something that may be really quite interesting. So we started to think about what the mechanism would be. And this, to be fair to you guys, this is still a work in progress. We've been working our way through. It took us 15 years to figure out the microglia signaling, and it wasn't just our lab. Right now, it's only our lab working on this. So one of the things we thought is maybe there's something neuroimmune going on, but not in the spinal cord, but in the periphery, in the nerve around where we've been looking. So we took a shot at a couple of different cell types, and F480 is a way to stain macrophage monocytes in peripheral tissue. And so you can see very little staining of macrophages here, lots of staining. In this case, the nucleus pulposus would be around here, and that to a very gross degree, the amount of F480 staining matched temporally up to the maximum when the behavioral sensitization was maximum, and then falling along with the decline in the hypersensitivity. So got us thinking that it might be macrophages that's involved in this. And so we did a couple different kinds of experiments to try to address that. One is, and these are just, we just took these as tricks from people that work on macrophages, peripheral macrophages. One is to inject claudrinate liposomes before, and we showed that this was able to essentially completely obliterate circulating macrophages. And what we find in those animals that had received claudrinate liposomes a few days before the nucleus pulposus surgery was that the behavioral sensitization was completely prevented in those animals. But in spinal, spared nerve injury, this depletion of peripheral macrophages had no effect on the behavioral sensitization. Again, a dramatic opposition between traumatic nerve injury and this non-traumatic type of nerve injury. We also use selenium, which some of you may know. The way I think of selenium as a non-immunologist is this is a way to kind of bash macrophages and stop them from working. It also has some effects on neutrophils. That's been recently reported. But it does interfere dramatically with macrophage function. And again, applying the nucleus pulposus in animals that are being treated with selenium, no hypersensitivity, but continue to have hypersensitivity in SNI. So this remarkable divergence of mechanism between traumatic nerve injury and this kind of atraumatic nerve injury. So we've been continuing on this, and it is definitely a work in progress to try to understand what's happening. So how is it that these microglia are able to change behavior at all? Like, to me, it's quite a bit of a mystery. And I'm not going to go through it, but we have quite a bit of evidence now around a couple different potential mediators that can be released and we feel act locally must change the excitability of the axons of passage as they're going there. So the model is, and I haven't shown you any evidence for this, but we have evidence for the nucleus pulposus cells have to be alive. The reason that for the reversal is probably the death of the nucleus pulposus cells. As long as they're alive, they're probably releasing soluble factors that we haven't identified yet, but we hypothesize are there from several in vitro experiments that causes the production of some one or more mediators that can act on the nerve to change the way the inputs are to the spinal cord and drive this sensitivity. Now, how does this fit with this other model that I was telling you about that we've been working on for so long? So the key to the other model is the immune side and the neuron side. And the neuron side involves some different kinds of receptors signaling to drop KCC2 or enhance NMDA receptor function. And we have ways of interrogating this. So we can do this with this compound CLP, which Yves de Koninck's lab identified, and APV. So this facilitates KCC2, this blocks NMDA receptors. And what we find is in the nucleus pulposus model, systemic delivery of CLP reverses the hypersensitivity produced by the nucleus pulposus, as does aminophosphorylvaleric acid. So the idea then we have from this is, until a couple of years ago, this was our model for sort of the grand unifying theory of everything, at least in our hallucinations. Now we need to modify this model to say that there is at least one other way to get from the naive state to this hypersensitive state. And the key to this is this change that happens in the neurons. These are neurons, and I didn't tell you this before, but these are neurons which project from the spinal cord to what you could call pain centers or pain pathways in the brain. As Yves de Koninck says, when these neurons fire, you say, ouch. Okay, so that that's where this is now. And if I have a couple more minutes, maybe I can take you through. So we're continuing to work on this because there's lots of questions. As I said, Tuan apparently has sorted this out. We're still trying to figure out how this happens. And my view of this, I told you about the nucleus pulposus at the beginning, that we were interested in getting a disc herniation model. I think what we've actually come up with is a neuritis model. And that certainly makes the case that you don't have to have microglial reactivity or microglia involvement at all in the spinal cord to get neuropathic pain hypersensitivity, at least in rodents, okay? So the other side is to tell you the work of Shahrzad Ghazisieti, and if you want, we're going to talk a little bit more after. Ask me how Shahrzad got to my lab. Okay, it's an interesting story about Donald Trump. Okay, so Shahrzad came to my lab through these circumstances, knowing nothing about neuroscience, being a ovarian cancer biologist and interested in sex differences and everything. And so she ended up coming to my lab, and she made a very compelling case, and I couldn't say no. Turned out to be one of the best decisions I've ever made, because she came with some ideas that have turned out to, again, give us a very different twist on things. And so part of what she had done was to sort of survey all the areas where there are sex differences and sex similarities in sort of peripheral and spinal pain signaling pathways. And this is in a article that she published last year. And I'm not going to go through all of those, just to say this is what I was sort of telling you about over here. But there are certainly other areas where there are sex differences in signaling or sex similarities in the peripheral endings, which aren't shown here at the site of nerve injury. This is for neuropathic pain, and in the dorsal root ganglion. So lots of different opportunities there for sex differences. And when she started the project I'm going to tell you about right now, the idea was we had been working on the sex differences, right? This was around the time we were simultaneously working on the nucleus pulposus project. Shahrzad came to the lab and we were working on this. So we knew that there were sex differences. And for a long time, I became obsessed with sex differences. And so was Shahrzad. She was obsessed with sex differences. So the idea was, can we find drug targets that are sex different, but species conserved? Because if we could find two species with the same target, maybe that would help in prioritizing drug discovery, because we have, many of you think mice and rats are similar. Most mice and rat people think they're very different species. They are definitely different species. So Shahrzad set up this, what I think of as kind of a heroic experiment, to try to use old school bulk RNA sequencing to see if we could pull out any information that might give us a hint about drugs. And in particular, already FDA approved drugs. And so the idea was, despite, all this fancy stuff that you guys may have seen already or maybe not around single cell sequencing and every tissue, it's single cell this and single cell that, it's like, but wait, can we get anything useful? So this was, for me, a proof of concept. This was entirely Shahrzad's idea. It was, for me, proof of concept to see if there's any utilitarian value to going old school, okay? And part of the appeal of this is that there might be a number of different control groups. So this is the experimental design, rats and mice, males and females. These are all the experimental group. And then these ones are groups that we propose to consider might be comparator control groups, right? 3 to 1, okay? And as it's turned, you're going to see in the next few slides, as it turned out, the power of doing bulk sequencing was dramatically enhanced by having a large number of animals in what turned out to be bona fide control groups, okay? So the first part of this, the analysis to look at this, was to do principal component analysis in mice and rats, which very clearly showed different principal components for the experimental group, which are the triangles. They cluster independently of all the three potential comparator groups. Another way to look at this is by looking at differential gene expression. And those of you that have ever looked at these will say, those are the most boring volcano plots I have ever seen in my life. Why are you putting them up there? Right? Because they're boring. No differences. Nothing came from this, okay? And then another way to think about this is non-supervised hierarchical clustering analysis of the transcriptomes from these various animals. And the main clusters then fall out as the experimental groups. That's the ones indicated by gold here. And then the other groups, sham, contralateral, etc. They fall out over here, okay? So by the time we got through this, we convinced ourselves, and actually reviewers, that we could make bona fide comparisons between the experimental groups, the nerve injury groups, and these control groups. And as I said, I think that's part of how this came about to give us some interesting information. Okay, so then we start then comparing the control groups to the experimental groups in a variety of ways, and then the volcano plots are not so boring. We get a lot of different differentially expressed genes, and we see those in comparing males and females in mice. They cluster together, they correlate. There's still some outliers, so maybe we've got some sex different ones. Same thing in rats, and there's a good correlation in mouse and rat, which would be part of the idea that maybe there are species conserved sex different genes. Okay, continue on. Start looking even harder now that we've gone through all this sort of prep work on the analysis. So I don't know how many of you have ever looked at a 2x2 Venn diagram before. I had not until this, and I think it took Sharzad a month to convince me how to understand this, right? So remember, we started off thinking we're looking for sex different species conserved genes. Now, probably you guys can figure it out, but she had to point this out. This is where they would be. So she found many, couple hundred differentially expressed genes in all these comparisons. But here's the number here, and here's the number here, and there's nothing of substance. 4 out of, I don't know, 250 different genes, more than that, 300 and something. Right? So that got us scratching our heads a little bit, at least me. And then after looking at this Venn diagram multiple times, I think Sharzad figured it out. Immediately, we saw this staring us in the face, right in the middle, sex conserved, species conserved genes that are changed with this bulk RNA sequencing. It's like, ooh, okay. Once again, observations tell us we got to look at what we're thinking about critically and think differently about this. So what do you do with genes like this and you're thinking like Sharzad? Well, you can take those 93 conserved differentially expressed genes and look at the proteins in them, and because drug targets are going to be proteins, right? They're not going to be genes, at least the way we were thinking about them. At least there isn't an FDA approved drug that directly acts on DNA. At least in this context. Okay, so pull out those, generate using this approach with a program called String, trying to figure out the potential interactions of all these genes. Most of the 93 are represented here. And then if you're interested in network theory, you can go on to calculate something that I had never heard about before called the integrated value of influence. So it figures out not only the connectivity, but the strength of the connectivity. And then that's represented here by the color and the shading of these. So you can see, well, genes or proteins with large integrated value of influence are in the darker and bigger, and those ones with less are less. So this is kind of sort of relatively run-of-the-mill network analysis for protein-protein interactions. But the insightful part was to ask, can we use this information to construct some kind of ranking of FDA-approved drugs? Is there something that comes out of this? So Shahrzad knew about this thing, which is called the drug gene database, a publicly accessible database. Any of you guys heard of this before? I had not heard of this before. It's publicly accessible, 10,000 genes, 40,000 drug gene interactions, and hundreds of thousands of other interactions. So basically, Shahrzad took the integrated value of influence, convolved it simply by adding up the numbers and creating a list of genes or proteins that are high on both lists, right? And so you can create a list of all 93 of the genes in rank order, and then we look and ask, okay, so which of these drugs that's FDA approved is on the top? And it's this drug. You've heard of this? No, nobody. Used for idiopathic, FDA approved for idiopathic thrombocytopenia purpura, right? Never, ever linked to pain. So I thought, oh, that's good. Okay, but there is a small but. Remember I told you this is a proof of concept study. If we're getting something that's meaningful, it has to act somehow and reverse pain, right? That's the idea. So take that approach. So it turns out fostamatinib is actually a prodrug. It's orally bioavailable. It's converted in the liver into this drug called R406. And so the experiment then was to ask whether if we administer to R406 to animals that had received the spared nerve injury, could we reverse the hypersensitivity? That was the whole point of the experiment, right? So this was 3 or 4 very tense weeks, first trying to figure out how to solubilize this stuff, because how to solubilize this, because it's not very soluble. It's greasy, right? And then figure out how to do the controls and do all the blinding. And half the people in my lab are now working on this to do the blinding correctly, do all the control experiments, figure out how to do this. And of course, I'm not going to be telling you this story this way unless this happened, right? The hypersensitivity is reversed by intrathecal injection of R406. Okay, so that then ended like a couple of years and a very tense month for the lab, because we're all wondering whether this proof of concept was ever going to get us anywhere. And so we got this far. Now, turns out that for ITP, there was a very specific target that was identified by all the manufacturers that were looking at this, which is spleen tyrosine kinase, or abbreviated SYK here. So this is the structure of SYK. This is a high power view of R406 bound to the catalytic spot in SYK. So now, Flavia, I know you've been paying attention, and you didn't see SYK on that other list, right? Nobody saw SYK, right? Just say yes. Yes, no, it wasn't on the other list. So how are we getting this? Well, turns out to get that, we were so focused on getting sex conserved, sex different species conserved, that the first pass at the network analysis was done with a really, really high cutoff. So we just tweaked the cutoff a little bit, and this is the network that you get. And so the biggest thing with the biggest integrated value of influence is SYK, right there. Something that had been totally missed in all the RNA-seq single cell stuff that had been done in the spinal cord, missed or ignored by everybody. So, okay, well, if it's true that SYK is the target, the molecular target for this, then there's a couple things should happen. We should find less SYK mRNA, and essentially, we have the transcriptomic data. I think the cutoff was so stringent that one animal in one of the groups caused SYK to be eliminated from the first pass. So we went back and looked at mRNA level by RT-qPCR, up in the pain groups in both sexes compared to the controls. And then, of course, SYK protein is actually the target, not the gene. So SYK protein is up in both sexes in both species. So, okay, so we're getting a little bit more confident, but I did tell the students before that my first postdoc mentor, John McDonald, had many rules of thumb, and the one I like the best is his rule about drugs. And his rule about drugs is all drugs have two actions, right? The one you know and the one you don't know. So, okay, R406 is a Syk inhibitor, but maybe it does something else. The other rule I have in my lab is that every molecule in the world is an NMDA receptor blocker until proven otherwise. Because NMDA receptors have so many allosteric sites and are inhibited by so many things. Okay, so what do we do? Well, it turns out ITP seems to be a good disease where drug companies are interested in. So there are a number of other compounds that are very good for ITP. This is one of them. It's not a prodrug. It's called P50515. You need to remember that because there is an exam afterwards. Structurally dissimilar from R406 and produces the same thing, right? This reversal of hypersensitivity after intrathecal injection. Okay, so now we have two structurally distinct drugs. They both reverse hypersensitivity for this target that we would have never thought about except if Charzad hadn't done the experiments in this way and gone old school with bulk RNA sequencing. Turns out Syk is very well known in immunology as an adapter protein for many different cell surface receptors. And so we've started to look now at the signaling pathways that might be involved and how this is working. Fortunately for us, there are a lot of mutant Syk mice that are out there. Well, SYK mice, not SICK mice, Syk mice that we can use, especially from a person who's now our collaborator at the University of Virginia and studies Syk in the context of Alzheimer's disease. But for me, the biggest question is, where does it go in here, right? This is my continuing hallucination about how things work in the spinal cord. Where is it, you know? And the most obvious place is here, except there's a sex difference here and there isn't one there. So Syk is expressed in a lot of different immune cell types. So maybe it's Syk in both and we just stumbled upon it this way, or maybe there's neuronal expression of Syk and it's here, certainly not over here in this model. And we gave it intrathecally, so it has to be acting locally in the spinal cord. So this is the biggest thing we're trying to work out now. Cell type here, and mechanism, which Syk is being activated, it's increased, is it being activated? What are the upstream regulators that are activating Syk? And then can we, give Syk inhibitors IV? Are there other things that we need to know about? So that's the end of the story. The two stories is that we've come to this kind of model about, or cartoon about where we think things are, and we're continuing on to try to find something. Now we've got something as a Syk target, maybe something that would be broadly useful. And obviously for the microglia independent form, is there any action over here? So lots of work to do, but we're still working through it. So it's been a good ride so far for the last 42 years. So, okay. These are some of the people that are involved in my lab and many other labs over the last number of years. And really, this is the lab. And for Charzad's story, we had a lot of collaboration with the Center for Computational Medicine at SICKIT. So I'll end there and say again, thank you. Thank you, Flavio, for the chance to come and visit the school and catch up with you again. Okay, ciao.
So I would like to thank Professor Solter for this interesting lecture, even to open a new option to understand the mechanism that, in my opinion, could be much more related to macrophages activity for the CYK, because maybe it's related to the chemotaxis of the macrophages.
Yeah,
maybe.
We got lots of work to do.
Yeah. Okay, so please sit.
It's okay if I leave that up there in case anyone has questions.
Yeah. So this
To throw at it, feel free to throw arrows.
Yeah. So the session is open for discussion. So I invite the students, because you have several interesting points to ask on. No question?
Already interrogated.
Okay, come, Maurilio. Yeah, but I don't know if they have seen this data before the line.
No, I gave a hint, a little bit of a hint about.
Okay. So introduce yourself and
Okay, hello, I'm Maurilio Menduni De Rossi, a third-year medical student here at Sant'Anna, and thank you for this lecture, very, very interesting. And I had some question that focus mainly on the sex differences, so the nerve injury model. And first of all, you said that these kind of sex differences were two different paths that that can be switched between sexes. Are they switched from like hormonal levels? And if so, in humans, for example, when someone starts to take some hormones, for example, in a transition medical experience, would they change this kind of pathway?
Yeah.
And in this regard, what could be the evolutionary reason for these two kind of different pathways?
Yeah.
And last thing about this, could this be used clinically, for example, to kind of, if I changing this pathway could change the phenotype of pain?
Yeah. So I'm going to start with the end because just to reiterate the point that we don't see phenotypic differences in the behavior, it's molecular differences. But
Okay.
the idea of being able to, if we miraculously had a drug for the part of the pathway that's in males or females, can we switch the other sex to also be sensitive? So I'll tell you, it is a hormone story, okay, the difference. So I'm going to ask you guys a question, which is it testosterone or estrogen? Who says estrogen? Okay. And who says testosterone? Testosterone. Yeah, but you read the paper. You're cheating. Okay, so it turns out that we've done quite a few, but not an extensive amount of characterization of this. And the next part of the story, which was in the original paper, is about using gonadectomy in males and females and using minocycline, which I told you I hate, but using it as a probe for microglia dependent pain, right? So in otherwise naive females that had males and females that had that had SNI, minocycline reverses the hypersensitivity in males, not in females. Gonadectomy doesn't affect the lack of effect, doesn't change the lack of effect of minocycline in the females, but prevents microglia from being active in the males. Give testosterone back to males or to overectomize females, and they both become sensitive to minocycline. And so the idea would be that it's testosterone levels in the males that are driving up the sensitivity to minocycline, and by inference then that it's permitting the pathway that is microglia involved. Okay, so how does that work? So it turns out that P2X4 receptors are activated by what you could think of as a small transcriptomic pathway that goes through true transcription factors, IRF8 and IRF5. And so just as another career advice, when we had this story originally, sorry, I'm digressing for a second just to give you guys a suggestion. So when we had this pathway, it was, me and Eve and Kazu. And so Kazu and I decided, Eve was willing to work on the neuron science. Kazu and I decided that we kind of divide the world into before the neurons and neurons and after, okay? So we worked out, the signaling pathway, and we continued to work with Eve on ferreting out a bunch of things. Kazu worked on what turns on P2X4 receptor expression, basically has been the major thing he's been interested in, and identified a bunch of signaling events that can do that under various conditions, and that there is this IRF8, IRF5 pathway. So immediately we thought, well, maybe the IRF5, IRF8 pathway is not on in females for some reason. So again, we went old school, QRT-PCR from the spinal cords, IRF5, IRF8 up identically in males and females, but P2X4 receptor message down. Okay, so IRF5 binds directly to the P2X4 promoter. So it's something to do with transcription that's not allowing P2X4 receptor message to go up in the females. The story then turns back to Sharzad because her other project was also massive. And she did a methylation study of changes in DNA methylation in spinal cord, periaqueductal gray, and blood. So we had some ideas that there may be similarities and differences. And she found lots of differences. The paper is this, I don't know, it's got 80 figures or something. It's going to be crazily massive. But as part of this, I don't think I have it on the stick, but she found many different differences in DNA methylation. So sometimes supervisors do add value. Okay, so I asked her one question about that because I knew nothing about DNA methylation. I said, is it kind of random, the differences in DNA methylation? Are they randomly distributed across the genome, or are there areas where there's more or less different? So it turned out that she identified a couple of what we've started to call hotspots for differences in DNA methylation. And one of them is on mouse chromosome 12 in a region on which lies P2X7, another P2X receptor, or A1, and a number of different microglia genes, including P2X4. So she looks at male and female, finds out that there's actually two binding sites in the P2X4 promoter for IRF5. They're methylated in females, not methylated in males. So I think that's ultimate. Now, why is that? How does that come about? Sure. But I think that's ultimately the difference between males and females. For some reason, in females, there's methylation of those two sites compared to males. And that happens before the nerve injury, right? So it's not induced by the nerve injury. So that...
Yeah, I can add something because estrogen activate the DNA methyltransferase 1. So that's why you have a higher magnitude of methylation in females.
There, but not all the methylation is higher in females. There's lots of methylation in spinal cord blood.
Yeah, maybe because it's related to the amount of oxidative stress.
Yeah.
Because there are some demethylase enzyme like the TET, that are related to the presence of oxygen, even the presence of oxidative stress. So if this TET, maybe there are some zone with higher oxidative stress, and so you can condition the demethylation of some area.
Yeah. Okay, good. We've got some things to...
It's related to the alpha-ketoglutarate, the Krebs cycle.
Oh, yeah? Okay. Yeah. So does that fit in? Doesn't come to mind how that would fit in directly from work that other people have done, but okay, that's good. Thank you for that suggestion. Yeah. So did that answer all your questions?
Kind of. I was just curious about also what you thought about the reason of these differences. Why should this specific model of chronic pain and not the nucleus pulposus, for example, have sex differences? What could be the evolutionary advantages of such processes?
Yeah, so I think generally there is survival value, right, to being able to detect pain. If you think about how much trauma do you need to damage a peripheral nerve, it's much more than, maybe breaking bones would be one thing that could do it, or, very severe trauma. So these are like stopgap measures for the nervous system to take to be able to respond, that you could think about it from that perspective. The other way that I think about these changes is, at least with the traumatic nerve injury, if you cut axons along the neuraxis or in stroke, there's a lot of microglia reactivity, on the cells that are injured, right? And nobody really understands how that is. It's probably related to CSF1, at least in the spinal cord. But originally we thought we're going to cut the nerve. We see this big microglia response. We're just going to trace all the tracks through the entire CNS, and we're going to find pain centers here and pain there and everything. And so it turned out there's only one other place in the central nervous system that we found exuberant microgliosis, which is in the dorsal column nuclei. So any of the students remember your neuron, maybe you haven't learned neuroanatomy yet. What's in common between the spinal cord dorsal horn and the dorsal column nuclei that relates to primary sensory neurons? Anybody remember? It's a tough question. I know because I was a pain biologist when I was a student, but spinal cord dorsal horn and nucleus pulposus, gracile nucleus or dorsal column nuclei are the termination sites of large fibers, A beta fibers, the primary termination. So at the ends of where the A beta fibers are, you see microgliosis. So that's been our working hypothesis. We've been distracted with a lot of other things, but we think that's why. It's a response to nerve injury. If you look in stroke, then where the neurons are, they get a microglia response, but it doesn't jump synapses. It's just on the cells that are damaged. And that's, you may have noticed in some of them you saw in the ventral horn, this massive green signal or a massive pink signal. That's where the motor neurons are. Mm-hmm. So those are neurons whose axons have been cut as well. So we think it's a response to axonal damage, and maybe then provides some advantage in terms of having remodeling of that take place, right? That gives you plasticity for repair and for other things that might be useful when you have a neuron that's damaged, that the processes respond by secreting something or somehow telling the external environment, hey, I'm damaged now. So I think that's a commonality between stroke and nerve degeneration, and spinal cord injury is the same. Yes. Yeah. For the primary afferents? Right. Yeah. Right. Almost all the A beta fibers that ascend in the dorsal columns, ipsilateral. Yeah. So that's why I don't know. We don't know anything more than that, but I think there's got to be some value in this. Having damaged the endings, you could think about the changes that we see: loss of inhibition by this mechanism, excessive NMDA receptor activity. Those are plasticity promoting functions. Even, in development, KCC is low, chloride's high, and at many synapses, GABA's excitatory during development. So there's lots of plasticity. So you get damaged, you promote plasticity mechanisms, and then you rewire in the adult. Mm-hmm. It's just when that happens during development, it's okay. When that happens in the fully developed nervous system, where the output of those neurons that are next goes to pain centers, then it's not so good, right? It's good for rewiring, but now you've got enhanced throughput through pain pathways, which you don't during development, right? So that's why it's useful. That's why we end up with this. It's a side product of the biological process of increase in plasticity in response to nerve cell injury. Thank you. It's a bit of hand waving.
Okay, wow. Okay, come. So epigenetics is really important.
Yeah.
So thank you for the lecture. I'm Antonio Raffaele Citarella, second year student. And I was wondering whether microgliosis, we saw that maybe it inactivates KCC2 at a peripheral level. I was wondering whether it also impairs GABAergic system in the brain, and if there is some parallelism between what happens down there and in the brain.
Yeah. So one of the potential mechanisms in some forms of epilepsy seems to be BDNF-driven disinhibition through this KCC2 downregulation system. So I think, again, under aberrant conditions, and there's been a lot of work on KCC2 and other systems, and there seem to be a lot of different pathways that converge on downregulation. So BDNF is one of them. In the brain, there are a number of other receptor-coupled pathways. A lot of GQ-coupled pathways can go that way as well as non-receptor tyrosine kinases. So I think that is a convergent pathway.
So do you think there is somehow a way to act on both pathways? I mean, down there and at the brain level?
Yeah, so in terms of the response to peripheral nerve injury, I think if anyone's looked in so the main projection site of the output of these spinal cord neurons is lateral parabrachial nucleus, at least in rodents, and thalamus. But the sort of pain-causing ones seem to go to lateral parabrachial nucleus. And I don't think anyone's looked there for KCC2 changes. But KCC2 is interesting because it's got a lot of advantages as a molecular target. Because the other, the sort of classic way to enhance GABAergic transmission is with allosteric modulators of GABA receptors like benzodiazepines. Yeah. They work, lots of problems with them. KCC2, if you think about it, I'm sorry, I'm digressing a little bit because I will come back to the question. So KCC2 is normally on in the adult brain, not so much during development, comes on and during development, on in the adult brain. So chloride is pretty much as low as you can get. So activating KCC2 more has very little effect on the level of chloride. So basically no effect on synapses, GABAergic synapses, that in cells where chloride is normal, right? So, but in cases where chloride goes up, KCC2 has gone down, chloride goes up, then there's targeted effects of KCC2 activation on those cells and those synapses. So basically, these are potentially drugs with minimal side effects, unlike benzos and anything else that facilitates GABAergic transmission. You're not changing GABAergic transmission, you're changing the postsynaptic inhibition. So Eve convinced me years ago that this is a great drug target. And you just have to think about it a little bit and look at the biophysics and say, yeah, he's probably right. So.
Yeah, and last question, do you think that microgliosis not only blocks KCC2, but favors the expression of NKCC, so the reversal effect?
Yeah, I mean, Eve's looked at that and they don't really see much. At least in the spinal cord, it seems that the main effect is on KCC2. We haven't looked at that, but we keep in contact with Eve a lot. So yeah, they did look at that.
Okay, thank you.
Oh. So guys, other question? Is clear? You want to suggest development of the hypothesis? No. So I have a question for you. So recently, I mean, in the last years, they detected some fibers in the niche of tissue, no? So, and people doesn't know what is the role of these neural fibers in the niche of a tissue. What is the role of pain in tissue regeneration?
In regeneration?
Yes. I mean, could be the pain the first hit to the regeneration of tissue?
So I think there's a couple ways to think about that. One is if you're thinking about acute injury and the effect of having a pain experience and learning that you should avoid this and resting. So resting to a certain degree certainly facilitates repair, tissue repair. As if you're continuing to use a broken bone, it's not going to heal very, very well. So there's that kind of action.
I'm talking about the cell crosstalk in the niche.
Yeah. So, yeah. But I think that's the biggest effect that helps repair is the lack of not moving the body part. The other part is there is some evidence people have, for example, cut primary sensory neurons. And that does affect, well, affects skin development, affects skin function. Trying to think of, I can't remember the exact paper, but does affect the ability of for tissue repair. So nervous, especially C fibers, so the small fibers that people would call pain fibers, but aren't really, have this function and efferent function. So I told the students that about Jeff Bernstock, the bombastic Jeff Bernstock, right? Pioneer of purinergic transmission. He regularly reminded us that C fibers are sensory motor fibers. They release lots of things at their peripheral terminals, including substance P, including CGRP. Migraine is involved in that. So people like Clifford Wolff and Isaac Chu have cut peripheral nerves and looked at regeneration, and regeneration and repair is dramatically affected by denervation. So there is some role, I don't think anyone's worked it out yet, for the efferent function, presumably efferent functions of primary sensory neurons. And we forget about that all the time. Even this pain school I was at, everyone forgets about these are efferent, can be efferents as well. We saw that, we demonstrated that at lunch, right? Neurogenic inflammation. That's an efferent function of C fibers.
Okay.
So a beautiful lecture. I don't know if I missed it, but did you explain to us the rationale for the use of nucleus pulposus, this autotransplantation?
Yeah,
why not a piece of liver? Why not a piece of bone? And second is just, and then I will finish. So are you sure that the effects that you see are not tissue specific? You have a control with another piece of, fragment of another type of tissue. Are you sure that there is no necrosis there? And so this is what attracts macrophages to the place?
Yeah, so we haven't put liver there. The rationale was very clear. I had a neurosurgeon working in my lab, that was his clinical practice, and he was trained as a bioengineer as well, or biomechanical engineer before, and had been working on, models where you get in there, dig around, and then, do something to nucleus pulposus or the discs, and you create all this mechanical trauma around the dorsal root ganglion. And we wanted to have as atraumatic as possible there. So we haven't put a lot of different tissue types on where the nucleus pulposus goes. But yeah, something I didn't show you at all, but I mentioned, is the nucleus pulposus cells are alive, right? So these are the cells. There are cells there, several kinds of cells that secrete the extracellular matrix that's making the gooey part of the nucleus pulposus. So we wondered, sort of along the same lines as you were saying, do the nucleus pulposus cells have to be there, or is it the matrix? That's the important part. And so we did a whole series of experiments where we killed the nucleus pulposus cells by ionizing radiation. So dead tissue, put it there, no effect. So yeah. So yeah, we wondered essentially about the same thing. We never thought about putting liver there, but that's kind of an interesting idea. No, yeah, I know what you mean. Yeah, is it just dead tissue there? So the nucleus pulposus cells seem to, certainly they remain alive, because we've done other experiments, because we were trying to think about whether, because the nucleus pulposus cells are motile, and we wondered whether they stay in the nucleus pulposus or whether they crawl out onto the nerve, and that's part of what's going on. And so we used GFP expressing mice that express GFP in every single cell, use disc material from them, put it on the sciatic nerve, and then looked for GFP and F480 staining. And it turned out that the nucleus pulposus cells all remained in the nucleus pulposus, whereas the macrophages were all around it, actually weren't really in the nucleus pulposus. Maybe they couldn't get in there very well because of the matrix. So that's where we got the idea that there needs to be action at a distance for the nucleus pulposus cells. And we did a series of in vitro experiments where we could get release of something. We still haven't figured out what it is, release of some diffusible factor that can attract macrophages.
You talk about sex difference. What about age-dependent difference?
Age dependency, yeah. So we've done nothing on that.
Nothing.
Nothing. And the field has done virtually nothing about that. There are a handful of papers in the pain field generally, the non-clinical research pain field, the sort of biological mechanisms. Almost nobody works on aging.
No one. So let's start, guys. Start to work about aging and on this issue and pain is a virgin, right?
Yeah,
Virgin
and
field.
certainly for, there have been a handful of studies, and there have been a handful of studies that have followed animals for a long time. So Jeff Mogul is one person who's done this and has related pain to shortening telomere length and things that prevent telomere shortening or prevent the very long-term.
That's
about it.
Yeah, it could be really interesting relationship with the phenomenon as a premature senescence also.
Yeah, well, that's what Jeff was selling for a couple of years.
Yeah.
Is that was the traumatic nerve injury are inducing systemically actually enhanced senescence, and he dropped that a few years ago. So I don't know if it's not true. No one else has picked it up at all. So
even in non-brain organs.
yeah, I think that's what he was thinking, was that there's a systemic response.
Systemic
The
response.
nerve trauma, and
Yeah.
that's causing widespread changes in telomeres and senescence. And he had some other measures of senescence.
So Lorenzo, is your time. Please take care about this advice and investigate on this issue.
You have to buy old mice and old rats.
Yeah.
That's
Yes.
the
Very.
problem. I think that's one of the major problems is
Yeah.
the cost of housing animals for that long.
So people use some fish to investigate aging, like the Notobranchus furzeri. No? Nervous system in fish, if you want to use as model compared to mammals and.
They have nociceptors. They have spinal cord equivalents. So a few people have started using zebrafish in pain research. They're just getting started, so they have to come up with a stimulus. How do you know when the fish will change their swimming behavior if they detect something noxious in the water. So I think they put capsaicin in the water with zebrafish, and they changed I saw there was one talk about this week at European Pain School. I'd never seen that before. So people are trying to use zebrafish as well. And there you can, the lifespan is quite a bit shorter, so you could more easily do aging studies.
Aging, yeah, of course.
Yeah. And then there have been some studies in fruit flies, not on aging though, but nociception in fruit flies. And there are a few genes pulled out from fruit fly screens. The main gene that got pulled out is TRPV1.
Okay. So if we have no more questions, I would like to thank again Professor Solter. A pleasure. And now the last part of our intense day will be some gift and even a picture and the book of signature at the end. Grazie a tutti e ci vediamo al prossimo incontro.
Great, thank you very much. No, this was fun. Great to bring this. Yes, great to meet you. Yeah. Good luck.